Hello everyone, myself Shivam Kumar. I am from BTech Biotechnology. I am from MIT University Noida, and topic for my study is investigate epigenomic factor landscape to categorize tumor for early diagnosis and patient treatment stratification. Introduction. As we all know, cancer is a very dangerous disease, and there are many research going around the globe to find a perfect cure for cancer that can fully cure a cancer. But only it's up to research only. And as you know, cancer is not a single step; it is a multi-level process, and they they might occur due to some external or environmental factors, or they can occur without any cause. Highly exposure of carcinogen to the body might cause cancer in the human body. Bioinformatics is a very important and helpful tool in analyzing cancer and treating. With the help of bioinformatics and traditional methods to cure cancer, it might be possible to fully cure a cancer in this cure a cancer. In this study, we will learn about various omics data to get hand on some tools that can be used to study cancer. There were three courses required to study to complete my project. Those were genomics, epigenomics, introduction to bioinformatics. Introduction to bioinformatics. Techniques like precision medicine and personalized medicine help in improving healthcare system around the globe. Molecular diagnostics detects specific sequence in DNA or RNA associated with disease. The finding of molecular features that could be used in diagnostics is often done with the help of bioinformatics. The use of big data to discover new medicine is still in early stages. Bioinformatics offers a wide range of possibilities from genomes and transcriptomics for target identification to proteomics and structural biology for small molecule optimization. We can optimize treatment regimes from the start by applying bioinformatics instead of going through the costly and time-consuming trial and error discovery process. Bioinformatics is an interdisciplinary discipline that includes biology, computer science, statistics, and more. As discussed in Introduction to Big Data Bioinformatics, we discussed big data informatics and its application in fundamental research, healthcare, and businesses. Bioinformatics is also helpful in agriculture. Agrigenomics attempts to find new solutions to challenge arising from environmental stress such as climate change, while also promoting the development of sustainable productivity for the world's growing population. The importance of genetic engineering and biotechnology enhanced breeding in culture is one of the most significant contrasts between agriculture and bioinformatics application. Plant breeding includes altering native genes, whereas genetic engineering involves in inserting a foreign gene into an organism. It resulted in better yields, reduced pesticides, drought resistance, and improved nutrition. Among other advantages, engineered organisms for features like pest resistance and nutritional value necessitate a thorough grasp of target organism genome and the use of foreign genes as a starting point. Introduction to genomics. A genome is a collection of DNA that makes up organism genetic material. Genomics is the study of genomes, which includes DNA sequencing and data analysis techniques. The size of data files, including genomic sequences, is frequently in gigabyte range. Bioinformatics has focused on the development of computer methods for the study of genetic data. The massive data element of genomics sets it apart from genetics. In most cases, genetics entails sequencing and researching specific genes and their function in inherited traits. Because the sequencing and analysis of complete genome present significantly different technical and computational problems than genetic genomics stand out. There are three types of genome sequencing. The first one is targeted genome sequencing. Targeted sequencing offers us with excellent coverage as well as a detailed look at what's going on in that particular location. This type of specialized sequencing could be useful if you wanted to find mutation that causes a specific condition such as cancer. With 40 to 400 genes, targeted sequencing works best. Targeted sequencing is suitable for examining a small particular section of genome in greater detail. It gives a gene a lot of coverage and lets us see single base changes like insertion and deletion. Targeted sequencing also creates less data, which may make analysis easier than with some of the large scale sequencing approaches. Whole exome sequencing. We can see all the gene changes with the whole exome sequencing from single point mutation to diverse insertion and deletion. Although entire exome sequencing gives less coverage than targeted sequencing, we learn more about a genome coding region. Whole exome sequencing provides us with a wealth of information, which is both a blessing and a problem. Whole exome sequencing, a complete genome sequence of 3 billion base pair, which is comparable to whole exome sequencing. This is significantly more than an entire exome sequence can produce. WGS generates a more detailed look at the information in our DNA by reading both coding and non-coding sequences. NGS, next generation sequencing. It is a massively parallel sequencing technology that offers ultra high throughput, scalability, and speed. It is used to determine the order of nucleotides in the entire genome, the target regions of DNA or RNA. There are two terms related to next generation sequencing. Those are variant calling and sequence alignment. Variant calling, the variation in genome sequence with respect to a chosen reference sequence. Sequence and alignment, to identify mutation, we need to match the read to the reference sequence. Epigenomics, the branch of biology which studies the causal interaction between genes and their products, which bring the phenotype into being. This system includes DNA methylation, histone modification, and non-coding RNAs are known to play fundamental roles in epigenetics. DNA methylation. DNA methylation is an addition of methyl group to the 5-carbon of cytosine in the DNA molecule. Histone modification are further mechanisms that regulate accessibility of chromatin for transcription machinery. Histones are proteins that pack an ordered DNA molecule inside the cell nucleus into compact structure called nucleosomes. And non-coding RNAs, the classical epigenetic mechanism include methylation of DNA and post-translational modification of histone. Epigenetics and disease. Methylation is a well-known and commonly used epigenetic modification pathway in cells. It has been linked to a variety of human elements, including malignancies, autoimmune diseases, and neurological disorders. Furthermore, methylation may be considered for complicated disorders influenced by secondary factors like as variation and age, which may alter the severity of the disorder. Dataset. Yeah, the dataset which was finalized for my study was of the hepatocellular carcinoma. In this paper, 64 cases of hepatocellular carcinoma was compared to 30 normal tissue, and there were various methods that were used to complete this study. The data has the accession number of GSE 87630. Now the final interpretation of raw data from dataset. So first of all, we downloaded the file in the raw mode, and after that the raw file was converted into an annotated file, and after that we performed various tests like t-test, p-value, t-test and find p-value and FDR and fold chain. We will identify down-regulated genes. For that, we need negative value of fold chain. And to identify upregulation, we need to find positive value of fold chain. We will sort the fold in descending order and then take out the top 10 genes in upregulated, and these top 10 genes are responsible for cancer. And the next step is we will sort the fold in ascending order and then find top 10 genes in downregulation and take out that top 10 genes that are related to the cancer. So here are some top 10 upregulated genes as well as top 10 downregulated genes. They are SPINK1, AKBR, 1B10, top 2A, CDC20, and many more. Thank you very much.